Can somebody confirm you can hear me?
I can hear you.
Thank you. I close my mail down in case it makes a noise. Excuse me if anyone heard that. Let me share my screen. Let's get out of there. Come on. All right, you should be seeing my screen now. Today is May 9th, and we should be discussing Chapter 5 and ending it. I don't think we will, and starting Chapter 6. Any questions about what we're doing? In the lab today, we'll be doing Lab 7, throat and urine cultures. All right, any questions? I guess I shouldn't shut that down. Oh, look at that. So I have that we were to here. We talked about the nutritional classification of organisms and stated you can classify them largely on what their carbon source is and what their energy source is. Let me shut my window in case car makes noise. There we go. So in energy production, nutrient molecules have energy associated with the electrons that form the bonds, the chemical bonds in the molecules. In cells, energy is obtained from catabolic reactions. So cells use catabolic reactions to get their energy. And then that energy is stored in the bonds of ATP. And then when the cells need the energy, they get it from ATP. This is what happens in most cases. ATP is the energy carrier of cells. It has a high energy and unstable chemical bond. And then energy can be released quickly and easily by breaking off the third phosphate group of ATP. Any questions about any of that? When energy is produced in cells, cells produce it by oxidation and reduction reactions. Oxidation reduction reactions are always paired, while one molecule or atom becomes oxidized and the other molecule or atom becomes reduced. Oxidation is the loss of electrons. Reduction is the gain of electrons. Oxidation reduction reactions are often called redox reactions. And what they did was they took the RED from reduction and then the OX from oxidation. And I don't know why they did it backwards, but oxidation reduction reaction is the same as a redox reactions. In an oxidation reduction reaction, we have two molecules or atoms, A and B. A loses an electron, which is gained by B. A is then said to be oxidized because it lost the electron. And then B is said to be reduced because it gains the electron. An exact example of this is you can have copper plus 1 plus iron, and it has 3 plus charges. In an oxidation reduction reaction, copper can become oxidized, lose an electron, and so copper will be 2 plus. Iron will gain the electron and will become reduced and will decrease to iron plus 2 charges. Any question about any of that? Oh, here's a how do you say that word? Mnemonic device. That's not how you say it. However you say that word, somebody can pipe in if you wish. I'm having trouble saying that word for some reason. For helping you remember oxidation reduction, you can say Leo the lion goes grr. So Leo, lose electrons, oxidation. Leo the lion goes grr, grr, gain electrons, reduction. Any question about that? And it's a mnemonic? No, that's not how you say it either. Ah, well, mnemonic, no. However you say that word.
I think it's mnemonic.
Mnemonic, there you go.
Mnemonic.
Yeah, I don't know how you spell it, but that's all right. Thank you. I know it was something similar to that. I'm just having trouble saying it. All right. Oxidation reduction reactions in biological systems. Oxidation usually has the proton removed with the electron, and then the proton follows the electrons. Now remember, in a hydrogen atom, it is composed of H plus ions, or a proton, plus an electron, and that's the hydrogen atom. And therefore, biological oxidations are often called dehydrogenations because the molecule losing the electron usually loses the hydrogen as well. And then the hydrogen just follows the electron. So let me blow this up so I can see it a little better. This molecule is going to become oxidized, losing the electron. It will also lose a hydrogen ion, which will then go to NAD plus. This is just an organic molecule like glucose, this one here. And then NAD plus is a carrier of the electrons, electron carrier molecule. And NAD plus takes the electron and the hydrogen and then becomes NADH. And that's oxidation reduction reaction in a biological system. Any question about that? All right. Sure, I missed something. Nope. All right, biological redox reactions or oxidation reduction reactions are used in catabolism to extract energy from nutrient molecules. And we're talking about most organisms do this, like humans, E. coli. Now, do green plants use biological redox reactions? Yes, they do, to get nutrient molecules. And they also can use catabolism to extract to get energy out of nutrient molecules. However, green plants can also do it in anabolism, and we'll talk about that later. The very last 2 slides or last 4 slides, something like that, of this lesson, I'll talk about photosynthesis. But we're mostly talking about animal cells and bacteria cells and getting energy from catabolism. You can get energy from anabolism, but that's only happening in chemosynthetic organisms and photosynthetic organisms. Highly reduced compounds, compounds that contain many hydrogen atoms, are degraded, that means catabolized, to highly oxidized compounds to give the cell energy. An example will be glucose, which is C6H12O6, and that has many hydrogens in it, a highly reduced compound. It will be oxidized to CO2 and water. Energy will be given off, and some of that energy will be trapped and then used to make ATP. Oh, yeah, excuse me. Any questions about that? Let me warn you, I usually sneeze twice, so let's see if another one comes. The point is, this highly reduced compound glucose contains a large amount of potential energy. And then when it becomes oxidized to CO2 and water, that potential energy, or at least most of it, is given off. And some of it is used to and trapped to make ATP. In our cells, this is mainly made in aerobic respiration. And aerobic respiration is one way that cells can make ATP. It is also called cellular respiration. I do not use that term because you can have respiration in cells like a anaerobic bacteria, and they are a cell, but they do not perform aerobic respiration. They perform anaerobic respiration. And when cellular respiration is used to mean aerobic respiration, I just think it's a confusing term. So I don't use that term, but cellular respiration does mean the same as aerobic respiration. In aerobic respiration, you have glucose, which gets together with oxygen, and then it gets oxidized, the glucose, in cellular respiration, giving off energy, which some of it is stored as ATP, and the glucose and the oxygen are converted to carbon dioxide and water. The summary reaction for aerobic respiration is C6H12O6 plus 6 oxygen atoms, or 6 oxygen molecules, plus 38 ADP molecules, and 38 phosphate, that's an inorganic phosphate, why it's PI, form 6 CO2 molecules, 6 water molecules, and 38 ATP molecules. Any questions about that? Now, this is a summary reaction where we're showing it in one step. In reality, aerobic respiration includes numerous chemical reactions and a few physical reactions. A chemical reaction is something where chemicals combine and then become other chemicals. A physical reaction is where a chemical doesn't change its form, it just changes its state, such as when you make ice cream, you pour whipping cream and milk and sugar in your ice cream maker, and then you turn it on, and the chemical reaction doesn't occur. The same milk and the ice cream and the sugar are in the product, the end product, but it's ice cream. And all it was a physical change where the sugar, milk, cream solution changed from a liquid solution to a solid that we call ice cream. And that's a physical reaction. Any question about any of that? So in energy production, cells generally make ATP, and some of the energy from some redox reactions are used by the cell to make ATP. When I'm saying some redox reaction is because human cells and many cells can only use 2 redox reactions to make ATP. All the other redox reactions do not make ATP, at least all the other reactions in our cells. And the only way we can make ATP is through aerobic respiration or through fermentation. And we'll talk about fermentation a little bit later today. And we have many other chemical reactions, which are, of course, a redox reaction, and they do not make ATP, such as the breaking down of sucrose into glucose and fructose. We can't use that reaction to make ATP. Okay? Any question about any of that? All right. So ATP is generated by phosphorylation to make ATP from ADP. Phosphorylation is just the addition of an inorganic phosphate group to ADP or some other molecule, meaning you can put a phosphate on some molecule and that's still phosphorylation. We don't have phosphate. Oh, there it is there. Phosphate shown right there. So to make ATP, we have ADP, that's adenosine diphosphate with 2 phosphates attached, plus energy. And then the third phosphate group is attached to make adenosine triphosphate, ATP, and that's adenosine with 3 phosphates attached. You'll note that both the second phosphate here and then the third phosphate is a high energy bond. And the cell can break these high energy bonds whenever the cell needs energy. Like when it, let me see if I have that next slide. Nope. Wrong one. Like when it needs energy for something, it can break off that third phosphate group and then harvest some of that energy due to the work that the cell needs to do. If the cell needs further energy, the cell could actually break off the second phosphate group from ADP and then use that energy for its energy needs as well. Any question about any of that? All right, so in the generation of ATP, cells have 3 mechanisms for making ATP. First, there's substrate level phosphorylation, and that's just the transfer of a phosphate group from one molecule to ADP to make ATP. So the phosphate comes off of this 3 carbon group and then goes to ADP, making ATP. Substrate level phosphorylation actually does happen in glycolysis, and we'll talk about that a little bit later, but we don't say that this is substrate level phosphorylation making the ATP at that point. We just say in this step of glycolysis, ATP is made. The second way that cells can generate ATP is by respiration. This is also called oxidative phosphorylation, and we'll talk a little bit more about that later when we're talking about the third step of aerobic respiration. You'll note that I'm not saying aerobic respiration here. I'm only saying respiration, meaning it's all forms of respiration, both aerobic respiration and anaerobic respiration, where you have the sequential transfer of an electron from an electron donor cell to an electron acceptor. Now, in aerobic respiration, the electron's donor is usually glucose, and we'll talk about it with glucose. And then the electron is sequentially transferred through an electron transport chain from one member of the chain to the other. And along the way, energy is given off. And then at the end, energy is released to generate ATP. And that happens in the third step of aerobic respiration, which can be called chemoosmosis. I'll call it the electron transport chain because it has many different names. But the point is, in respiration, when the electron is moving from one molecule to another, that electron gives off energy. And so the energy is not released all at once. And that way the cell can release that energy in controlled ways. And the temperature of the cell will not get so hot that it'll fry the cell. Because when you burn glucose, it gives off a lot of energy, bringing to CO2 and water, by the way. All right, any question about any of that? So the first way was substrate level phosphorylation. The second way is respiration. The third way is photophosphorylation. The third way is only seen in photosynthetic cells, where light energy releases electrons from chlorophyll. The light energy is then converted to chemical energy via an electron transport chain. ATP and NADPH are generated, but they are often used to make sugar. So in photophosphorylation, light energy is often converted into the energy to make sugar. And here we see a picture of photophosphorylation in process. That's why the boys are so muddy. Just joking. In the background, you can see plants which engage in photophosphorylation. So in energy production, the energy is released from molecules in controlled ways and stored by a series of reactions. The energy is not released in a single energy burst, because if that happened with glucose releasing all of its energy at one time, that would fry the cell. No question about it. In fact, that would turn the cell into char. That's how much energy would be released. So electrons from donor molecule are passed from one compound to another through a series of redox reactions. And it's not shown here, but the electrons leave glucose and then go to NADH. NADH then brings it to this molecule. The electron then goes to this molecule and then flows from the one above to the one below. And then in aerobic respiration, the electron ends up flowing to oxygen. That's why it's called oxidative phosphorylation or aerobic respiration. Most organisms use as their primary source for cellular energy glucose or some other carbohydrate, and then burn it to obtain their energy needs. And this is a catabolic process. Like I said, it is true that photosynthetic organisms and chemosynthetic organisms do not use catabolism to obtain their energy needs, but they can. We'll talk a little bit about that later, or maybe I'll just mention it now, and that is green plants, whenever they need energy, they can get it if the light is out from photosynthesis, but they can also catabolize glucose to get their energy needs, meaning make ATP and then use ATP for their energy needs. And green plants do that whenever it's nighttime, or in Point Barrow, Alaska, green plants do that for 3 months in the year, because in the wintertime, the sun never shines. For 3 months in the year in Point Barrow, Alaska, and further north, the sun never rises. And I guess in the summertime, I believe the sun never sets, but don't quote me on that last part. I think that's true. I might be a little wrong on the last part. I've never been in Point Barrow, Alaska. The most common energy source in carbohydrate catabolism is glucose. But an organism can use any other carbohydrate as long as they can digest it and then break it down into glucose and then send the glucose down the normal way in aerobic respiration. Organisms may also use other nutrient molecules like lipids and proteins, and you can burn them directly. Like a marathon runner can burn lipids, fats, directly in their marathon run. However, they don't do that until 45 minutes into their run. And before the 45 minutes of their marathon run, they are using carbohydrate catabolism, burning glucose or some other carbohydrate and breaking it down into glucose and sending it through aerobic respiration. It's not until 45 minutes into their marathon run that they'll send a lipid through aerobic respiration and then burn the lipid directly to obtain energy. So most organisms prefer using glucose for aerobic respiration. To produce energy from glucose, microorganisms use two general processes: respiration, which is both aerobic respiration and anaerobic respiration. And then the second general process is fermentation. Any question about any of that? If not, let's watch a little video on aerobic respiration. Assuming it loads. Oh, here it is. Nope, that's not it. It's not showing. Oh, that's not the right one. Sorry. There it is. Nope.
World where academic deadlines meet the blank page.
Grammarly?
Hi, yes. Graham,
There we go.
All cells need energy to stay alive. Usually your cells get their energy through the process called aerobic respiration. The word aerobic refers to oxygen. In aerobic respiration, oxygen interacts chemically with a molecule of glucose. The glucose is broken down into carbon dioxide and water. This process releases energy, which the cell stores in the chemical bonds of ATP. The first stage of aerobic respiration, glycolysis, occurs in the cytosol, the jelly-like part of the cytoplasm. Glycolysis breaks each molecule of glucose into two molecules of pyruvate, energizes two electron carrier molecules, and produces two molecules of ATP. The next two stages of aerobic respiration are carried out in the mitochondria. Pyruvate enters a mitochondrion. Once inside, it is modified and enters the Krebs cycle. The reactions of the Krebs cycle break the pyruvate molecules down to carbon dioxide, create two more molecules of ATP, and energize more electron carrier molecules. The final stage of aerobic respiration is electron transport. The electron carrier molecules that were energized in the earlier stages deliver electrons and hydrogen ions to the electron transport chain. As the electrons are passed down the chain, energy is released and used to make ATP. This final stage produces as many as 32 molecules of ATP from each molecule of glucose.
Any question about that? All right, this slide is showing you a summary of aerobic respiration on the left and then fermentation on the right. Aerobic respiration has three principal stages: glycolysis, the Krebs cycle, and the electron transport chain. Essentially, electrons flow from glucose, an energy-rich molecule, to CO2 and water, energy-poor molecules. Aerobic respiration has a high yield of ATP. In fermentation, we only have glycolysis occurring. There is no Krebs cycle, there is no electron transport chain. Instead, pyruvate is made by glycolysis from glucose, and the pyruvate is converted to the end product of fermentation, and that may be ethanol or lactic acid, lactate. It's whatever the end product is of fermentation. There is a much lower ATP yield from fermentation. An overview of aerobic respiration in a eukaryote cell, which was similar to what was shown in the video, although it didn't mention it was in a eukaryote cell, is, let me blow this up. The pink would be the cell, and inside the cytosol or the cytoplasm of the cell is where glycolysis occurs, where glucose is converted in a series of steps to pyruvate. Some energy is given off and is used to make ATP. The pyruvate then in a eukaryote cell moves into the mitochondria. That doesn't happen in a prokaryote because prokaryotes don't have mitochondria. The pyruvate is changed in the preparatory step, and then it can enter the Krebs cycle. The Krebs cycle is also called the citric acid cycle, and there's another name for it too. I'm losing it, what it is. TCA for the citric acid cycle. Oh, tricarboxylic acid cycle is the other name. I'm going to call it the Krebs cycle. Your textbook calls it the Krebs cycle after Dr. Krebs, who discovered it. And the citric acid cycle is, citric acid is one of the first molecules made in the Krebs cycle, why it's sometimes called that. In the citric acid cycle, you do generate some ATP, you do generate some NADH and FADH2, and actually glycolysis generated some NADH as well. The NADH and the FADH2 take the electrons to the third step of aerobic respiration, which I call it, and your textbook calls it, the electron transport chain. It can also be called oxidative phosphorylation or chemoosmosis. I don't use those terms because they're a little different from the electron transport chain, and it's just hard to remember all the different names for something. And this one has at least 3 names for it, actually 4, because you can call it the electron transfer chain as well as the electron transport chain. And those are so similar that most people don't get confused by that. Let me say that in the citric acid cycle, you do, as well as the preparatory step right here, you do generate some CO2, and that's actually where the carbon from glucose goes to. All of the carbon in glucose is burnt to CO2 in the preparatory step or the Krebs cycle. I'll talk about the preparatory step in a little bit. And then the electrons are carried by NADH or FADH2 from both the glycolysis and the Krebs cycle to the electron transport chain, and then they're used by the electron transport chain to generate ATP by oxidative phosphorylation. And this is a high yield of ATP in this step. Any question about any of that? Now, in prokaryotes, this doesn't happen in the mitochondria. The third step of aerobic respiration, or the electron transport chain is the third step, happens across and near the cell membrane. So it's happening across the cell membrane. Any questions about that? All right, the first step of aerobic respiration is glycolysis. And glycolysis has another name, which I don't use, the Emden-Merhoff pathway. It's the oxidation of glucose to pyruvic acid. This reaction occurs in most living cells. It can occur in the presence or in the absence of oxygen. It does produce a little ATP and some NADH. Here we're seeing glycolysis, a 9 or 10 step process where glucose, shown here, is converted into pyruvic acid, shown here. And this makes that molecule, which then goes to that molecule and then goes down. So you could draw it this way, or you could just break it this way and then have that with going down here. Although this will be a little longer because that will be converted to there in that step there. The important part is that 4 ATP come out of this reaction to here. Come on, mouse. To here. But you need 2 ATP to come into the reaction, one here and one here. So you get a net gain of 2 ATP generated from glycolysis. Any question about any of that? And I had to learn the names for all of the molecules and their intermediates, as well as the enzymes that performed each of the chemical reactions. And so I think it's fair that if I had to know it when I was an undergraduate, that you guys need to know all of these names for every step of glycolysis. What do you guys say about that? Think that's fair? Well, if nobody objects, then you have to know it all. The summary of glycolysis is you start with glucose, and then you have a net gain from glycolysis of 2 ATP generated, a net gain, 2 pyruvates, and that could be called pyruvic acid. The cell will treat pyruvic acid and pyruvate the same. Chemically, they are just different from whether the hydrogen is attached or not. For our use, we will treat them synonymously. A chemist would object, but who cares? Because the cell treats them the same. And then they also generate 2 NADH from glycolysis. Any question about that? All right. You don't need to know all of the different steps of glycolysis, but you do need to know everything in the red box of the summary of glycolysis. Respiration is the oxidation of molecules releasing electrons for the electron transport chain. Respiration has 2 ways of proceeding. There's aerobic respiration, where the final electron acceptor is oxygen, and there is anaerobic respiration, where the final electron acceptor is a molecule other than oxygen. It could be a nitrogen compound like nitrite or nitrate, or it could be a sulfur compound. It just depends on what form of anaerobic respiration we're talking about. The important part is ATP is generated by oxidative phosphorylation. And we're going to be talking about aerobic respiration. We will come back and talk about anaerobic respiration, comparing it to aerobic respiration. But in great detail, we're going to talk about aerobic respiration, which we are talking about. But anaerobic respiration does have glycolysis. It does have the preparatory step, and it does have the Krebs cycle. but the electron transport chain is a little different. Meaning it does have the electron transport chain, but it's a little different. In the preparatory step of aerobic respiration, the pyruvate is converted in one step so that it can enter the Krebs cycle. And pyruvic acid is converted into acetyl coenzyme A. NADH is actually generated. And then CO2 is given off. That's the first CO2 generated from glucose, meaning the carbon in glucose. Note the preparatory step runs twice because for one molecule of glucose, you generate 2 pyruvic acid molecules in glycolysis. You'll also note that oxygen has not been used yet, either in glycolysis or the preparatory step. The Krebs cycle is the next step. The Krebs cycle is also called the tricarboxylic acid cycle or the citric acid cycle, and both of these are abbreviated TCA. Obviously, that one's abbreviated Krebs cycle. In the preparatory step and the Krebs cycle shown here, let me blow this up to make it a little bigger. The preparatory step right here, generating NADH. The Krebs cycle shown here, where acetyl coenzyme A comes into the Krebs cycle, combining with oxaloacetic acid, making this molecule, which is citric acid, why it's called the citric acid cycle sometimes, but we'll call it the Krebs cycle, which is converted into this molecule, converted into that molecule, converted into that molecule, converted into that molecule. And this is where we make ATP in the Krebs cycle, converted into that molecule, converted into that molecule, and then converted back into oxaloacetic acid, which is why we call this a cycle. We end up where we began. So as long as you have pyruv or acetyl coenzyme A entering the Krebs cycle, this cycle will continue for as long as you are alive. Any question about any of that? As long as you have acetyl coenzyme A, the cycle will continue for as long as you are alive. You generate 1 ATP, 4 NADHs, 1 right here in the preparatory step, 1 right there, 1 right there, 1 right there. You generate 1 FADH2, 1 right there. And then you generate 3 CO2s, 1 right here, 1 right here, and 1 right there. That's for each cycle of the preparatory step in the Krebs cycle. But remember, for each glucose molecule, the preparatory step in the Krebs cycle will run 2 times. So if I ask you a question, how many ATP do you generate per 1 Krebs cycle? You would say 1 ATP. But if I were to ask you how many ATP you generate from 1 molecule of glucose in the Krebs cycle, you would have to tell me 2 ATP. So you have to watch how the wording is. Any question about any of that? All right, the NADH and the FADH2 carry the electrons, which ultimately came from glucose, to the 3rd step of aerobic respiration, which is the electron transport chain. All right, any questions about any of that? Let me state that once again, in a eukaryote cell, the Krebs cycle is happening inside the mitochondria. Literally, it happens inside the matrix of the mitochondria. It's inside the inner mitochondrial membrane. But the 3rd step of aerobic respiration, where the NADH and the FADH2 are taking the electrons, the electron transport chain happens across the inner mitochondrial membrane for a eukaryote cell. And for a prokaryote cell, it happens across the cell membrane. The electron transport chain is a series of carrier molecules that are in turn reduced and oxidized as electrons flow originally from glucose, and then they're passed down the chain of the electron transport chain. Energy is released as electrons transfer from the high energy bonds, and energy is used by chemiosmosis to make ATP. Let's watch a little video on the electron transport chain.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, two mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, two electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of one hydrogen ion for each electron. Next, the two electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. One hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
All right, any questions about any of that? So in eukaryotes, the electron transport chain happens across the membrane as the inner mitochondrial membrane of the eukaryote. In prokaryotes, this happens across the membrane as well, the electron transport chain, but it is the cell membrane. what happens is NADH and FADH2 donate 2 electrons to an protein in the electron transport chain. And it's a series of proteins. What's called the electron transport chain is it's a bunch of proteins in the membrane, and it's a chain of proteins. And the electrons flow from the first protein or the second protein to the lower, further down proteins. So in the first protein, the electrons go to it, and the electrons go to the second protein, going to the third, fourth, fifth, etc. And in this process, in some of the proteins, when the electron is moving through the protein, it gives the protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. And that happens here, here, and here. The electron will then leave the electron transport chain and enter the final electron acceptor. In the case of aerobic respiration, the final electron acceptor is oxygen. The oxygen combines with the electrons and with hydrogen ions to form water. And that's why we have to breathe. We have to breathe so that we gain oxygen to make water in the electron transport chain of aerobic respiration. Without oxygen, we cannot generate ATP, and obviously we would die. Okay? Any question about any of that? So all of the oxygen you breathe in is converted in aerobic respiration to water, shown right here. None of that oxygen is converted to CO2. Any questions about that? And I've told you where you get the CO2 that you breathe out. I mentioned that all of the carbons of glucose are burned to CO2, and that generates the CO2 that we breathe out. All right, in electron transport chain, we're pumping hydrogen ions from this side of the membrane to that side of the membrane. That will create a twofold gradient where there will be more hydrogens on this side of the membrane than on this side of the membrane. There will also be more positive charges on this side of the membrane than on this side of the membrane. That potential energy difference will be used to make ATP, meaning these hydrogen ions will, by the laws of diffusion, want to move from this side of the membrane to this side of the membrane. The trouble is they can't move through the membrane, except in this protein, and this protein is an ATP synthase protein. When the hydrogen ions move through this protein, they give that protein the energy it needs to generate ATP from ADP and inorganic phosphate, shown here. Any question about any of that? Let's see. So this is how ATP gets made in the third step of aerobic respiration, as well as how the oxygen gets used up and converted into water. Any question about any of that? So in the electron transport chain of aerobic respiration, it's very efficient at generating ATP. In eukaryotes, you generate 32 to 34 ATP molecules per molecule of glucose. Prokaryotes are a little more efficient. They generate 34 molecules of ATP per molecule of glucose. The heart, the liver, and the kidney cells of humans can generate 34 molecules of ATP per molecule of glucose. All the other cells in our body, such as your skin cells, your muscle cells, your bone cells, can only generate 32 molecules of ATP per molecule of glucose. Why do you think your heart, your liver, and the kidney are more efficient than your stomach cells or your skin cells? What do you know about your heart compared to your skin cells? Anyone? Well, let me put it this way. What do you know about your heart compared to your brain cells? Obviously, no one's commenting, so your brain isn't working. It's your heart is always working. Your brain is not always working. So your brain cell only generates 32 molecules of ATP per molecule of glucose, whereas your heart, your liver, and your kidney, which are always working, they generate 34 molecules of ATP per molecule of glucose. They're a little more efficient because those cells are always working. At least while you're alive, your heart is always beating. Any questions about any of that? All right. In aerobic respiration, the final electron acceptor is the molecule of oxygen, which combines with hydrogen to form water. And here we're looking at the electron transport chain and aerobic respiration on the left compared to fermentation. Let me go on there. And here showing you the electron transport chain where NADH gives up its electron to the first protein in the membrane of the electron transport chain. The electron then flows to the next protein, the next, and then it leaves the electron transport chain, and then the electron flows to oxygen, which will be converted into water. And this stage of the electron transport chain, you have a whole bunch of oxidation reduction reactions happening. Reduction is the gain of the electron, oxidation, the loss of the electron. Any question about any of that? This slide is showing you where the electron transport chain happens in a eukaryote cell. That's happening across the inner mitochondrial membrane, shown right here. And in a prokaryote cell, it's happening across the cell membrane right there. Now, in a eukaryote, the hydrogen ions are pumped from this side of the membrane to that side of the membrane. And so these hydrogen ions are in between the inner membrane and the outer membrane. And that will protect the cell from hydrogen ions. Any question about any of that? And then those hydrogen ions will flow back through the inner mitochondrial membrane at this ATP synthase protein. In prokaryotes, where those hydrogen ions are pumped, at least for most prokaryotes, if they have a cell wall, those hydrogen ions are pumped inside the cell wall. And then they flow through, once again, this ATP synthase across the cell membrane to generate ATP. Any question about any of that? I think I've already talked about that. This step of aerobic respiration of the electron transport chain is called chemiosmosis. I don't call it that. I just call it part of the electron transport chain. This is where the gradient of hydrogen ions is used to make ATP. The movement of hydrogen ion across this membrane through this ATP synthase gives that protein the energy it needs to make ATP. Your textbook talks about that. The electron transport chain is also called oxidative phosphorylation because phosphorylation is the combining of a phosphate group to a molecule, and we combine it to ADP to make ATP. And then it's happening in the presence of oxygen, why it's called oxidative phosphorylation. Like I said, I don't use this term because it's too different from the electron transport chain. So in aerobic respiration, the counting or accounting of ATP for eukaryotes, we generate 36 to 38 ATP in all of aerobic respiration. That's 2 ATP in glycolysis, 2 in the Krebs cycle, and 32 to 34 in the electron transport chain. In prokaryotes, you generate 38 ATP in aerobic respiration. And remember, the summary equation for aerobic respiration is C6H12O6 plus 6O2 plus 38 ADP and 38 inorganic phosphates generates 6 CO2, 6 water molecules, and 38 ATP. Any question about any of that? And all of these oxygens are converted into water. And then all of the carbon in glucose is converted into CO2. Now, this is telling you the numbers you need to know. All right, that's enough for aerobic respiration. Let's talk briefly about anaerobic respiration. Anaerobic respiration is where respiration occurs, but the electrons do not flow to oxygen. They flow to some other molecule, which we call the final electron acceptor. And it could be nitrogen, it could be a sulfur compound. It just depends on what form of anaerobic respiration we're talking about. And there are many different forms. I don't know how many, but obviously there can be nitrate, nitrite. I think it can be ammonia too. So there's 3 with nitrogen. There's at least sulfur and hydrogen sulfide, so that's 2. And I think there's ions. So there's at least a handful of different forms of anaerobic respiration. You don't need to know any of them except just anaerobic respiration, where the electrons go to some molecule other than oxygen. In anaerobic respiration, glycolysis and the Krebs cycle still occur. The electron transport chain also is there, but it is different. The electron transport chain of anaerobic respiration is truncated. So it is a little different. Because the electron transport chain is a little shorter in the anaerobic respiration compared to aerobic respiration, you have a lower yield of ATP. For prokaryotes, it would be someplace above 2 up to 36. And therefore, anaerobes generate less ATP per molecule of glucose than aerobes. So aerobic respiration is the most efficient way to generate ATP. But there are some forms of anaerobic respiration which are almost as efficient as aerobic respiration. And that would be the forms that generate 36 molecules of ATP. The poorest forms of ATP production and anaerobic respiration. I think the poorest is actually 4, not counting fermentation, which some people do call that anaerobic respiration. I'm not, because fermentation doesn't have the glycolysis, the Krebs cycle, or the electron transport chain. So I'm just going to call anaerobic respiration the forms of respiration that do have glycolysis, the Krebs cycle, and the electron transport chain. And I think the poorest is generating 4 ATP. I used to know what that was. I think it's the sulfate. Anaerobic respiration, but I might have it backwards. It might be the nitrate. Because anaerobes generate less ATP, they tend to grow slower than aerobes. So this is comparing respiration, both aerobic respiration and anaerobic respiration with fermentation. And you'll note that respiration, both anaerobic and anaerobic respiration and aerobic respiration, do contain glycolysis, shown here, the Krebs cycle, shown here, and the electron transport chain. Fermentation only has glycolysis. And let me go back a minute and show you what I mean by a shorter chain. Here's the electron transport chain of aerobic respiration. You can have this protein removed from the chain. Okay, and maybe that protein as well. And so the hydrogens are given to this protein here, and that would be a shorter chain. The very shortest chain, I think, only has this protein in it, of the electron transport chain. Oh, let's go back to where we were. All right, let's talk about fermentation. You do get ATP production in fermentation, but not very much. Fermentation does release energy from the oxidation of an organic molecule such as glucose via glycolysis. In glycolysis, you get 2 pyruvic acid molecules, you get a net gain of 2 ATP, and you generate NADH from the oxidation of glucose. In fermentation, you combine the first step of fermentation, which is glycolysis, to generate ATP, and then you apply the fermentation step or steps, depending on the fermentation process. It's either, I think, 1 or 2 steps in all forms of fermentation. There's only 5 or 6 different forms of fermentation, maybe 7, but I think 5 or 6. The very simplest is only 1 step. You convert pyruvic acid to lactic acid, but in all forms of fermentation, you convert pyruvic acid to the end product of the fermentation reaction. Fermentation uses an organic molecule as the final electron acceptor, producing a fermentation end product. In lactic acid fermentation, pyruvic acid itself is the final electron acceptor, and it will be converted into lactic acid. You don't need to know the final electron acceptor for fermentation, but my point is we do have a final electron acceptor, and that electron initially came from glucose. Flowed, that's the word I was trying to say. Which I guess flew is past tense of flown or whatever. All right, here's a question for you. In fermentation, we do generate some ATP in glycolysis. We do generate some NADH. The NADH is used here to make NAD plus. Why in fermentation would the cell use up this NADH in fermentation instead of saving it and waiting until aerobic respiration can happen, like in your muscle cells, which can engage in fermentation, by the way, lactic acid fermentation. We'll talk more about that in a minute. They could save up this NADH from glycolysis and then just wait until the heart and the lungs bring oxygen to the muscle cell. And then that NADH could be used by aerobic respiration to generate ATP. Why does the muscle cell use up this NADH in fermentation? Anyone have a guess? Anyone brave enough to comment? I'll give you an extra credit point if somebody comes up with the correct answer. 3, 2, 1. Nobody gets the extra credit point. The reason is that the muscle cell wants to have ATP so the muscle cell can keep working, contracting, and without fermentation, the muscle cell will not generate ATP, assuming there's no oxygen in the muscle cell. And so the muscle cell needs to use up this NADH to convert NAD plus so that the NAD plus will be here for the muscle cell to engage in glycolysis and then make ATP in fermentation. If there is no NAD plus, the muscle cell cannot engage in fermentation, cannot engage in glycolysis, and will not make any more ATP. No ATP, the muscle cell can't contract, and then the muscle can't work. And actually, every Olympics, usually we have one Olympic athletes who you'll see them running close to the finish line or something like that, and then they just collapse. What happens is their muscles used up all of the oxygen in the muscle cell in aerobic respiration to make ATP, and then the muscle starts falling on fermentation to make ATP, and the Olympic athlete continues running, then they're getting muscle burn from making the lactic acid in their muscle, and then they run out of glucose in their muscle. No glucose, no fermentation, no fermentation, no glycolysis, no glycolysis, no ATP being generated. The athlete then collapses. In the summertime, I think the coach runs out and sprays them with water. In the wintertime, the coach runs out and throws a blanket over them. And then the Olympic athletes, their heart and their lungs are working like mad to get oxygen to the muscle cells. The Olympic athlete will then step up and then walk across the finish line, meaning they're fine because just their muscles were running out of ATP. They couldn't work anymore. Any questions about any of that? All right, I've already mentioned that human cells, at least our muscle cells, can engage in fermentation. It's lactic acid fermentation that our muscle cells can make ATP, meaning they can use glucose to make ATP. They're the only cells in the human body which can make ATP from fermentation. And they only do that when the muscle cell runs out of oxygen And what happens is the muscle cell is using up the oxygen faster than the heart and the lungs can bring oxygen to the muscle cell. The heart and the lungs are trying, they're working like mad, trying to get oxygen to the muscle cell. But as you know, you get muscle burn. I'm sure I'm sure everyone's gotten muscle burn a little bit. And that's just because the heart and the lungs cannot get oxygen to the muscle cell fast enough, in which case the muscle cell falls back on lactic acid fermentation to generate some ATP so the muscle cell can continue working. Now, most of us are not Olympic athletes, and so we've never probably ever collapsed because of the lack of generating ATP. But like I said, usually you see one athlete, only one, each Olympic collapse because they ran out of ATP. Lactic acid fermentation is actually quite common. Obviously, our muscle cells can do it. Many bacteria can do lactic acid fermentation, such as the bacteria used to make sauerkraut, bacteria made to make yogurt, the bacteria made to make cheddar cheese and most cheeses. Most fermented food like tea, even chocolate, use lactic acid fermentation. Another common fermentation reaction is alcoholic fermentation, also called ethanolic fermentation, where glucose is converted to ethanol in 2 steps. So let me blow this up. Pyruvic acid is in 1 step converted to lactic acid in lactic acid fermentation. And alcoholic fermentation, pyruvic acid is converted into ethanol in 2 steps. And yeast can do this. The yeast for making leavened bread or making beer or making wine use alcoholic fermentation. So these 2 reactions you kind of need to do. No. The ethanol is a 2-carbon molecule. Pyruvic acid is a 3-carbon molecule. So CO2 has to come off when you're making alcoholic fermentation. And that's actually how beer gets its fizz, from the generation of CO2. In making of wine, it's made in an open barrel or an open casket or something like that, and the CO2 will vent to the air. But if you were to make the wine in a bottle and cap it so the CO2 would just stay in the wine, we call that champagne. Okay. And then in yeast making leavened bread, that's the CO2 being generated that makes the bread rise. Any question about lactic acid fermentation or alcoholic fermentation? There are some different end products and different fermentation reactions. Like I said, lactic acid is probably the most common. Alcohol and ethanol being the second most common. Propionic acid fermentation is the fermentation reaction used to make Swiss cheese. And CO2 is given off in propionic acid fermentation. And that's why Swiss cheese has a hole in it. There's also butyric acid fermentation, and I don't know what this one is called. And then this one of these is called acetic acid fermentation. I don't remember that one or that one. I think this is the one that is making the one that we use to make vinegar. Okay, but these are just 1, 2, 3, 4, 5, 6 different forms of fermentation. Any question about any of that? Here's some table. You don't need to know it. The different type of fermentation reactions. The starting material, you don't need to know that. There's Swiss cheese production right there. And the bacterium, you don't need to know that. Just a interesting table, I guess. This is interesting, and that is you can use this one to make vitamin C. All right, this is a summary of aerobic respiration, anaerobic respiration, and fermentation. Aerobic respiration happens aerobically in the presence of oxygen. Oxygen is required because electrons flow to the oxygen. But it differs from anaerobic respiration and fermentation. Oxygen are not used in anaerobic respiration or fermentation. Anaerobic respiration happens in the absence of oxygen, usually, because most anaerobes can't work in the presence of oxygen. There are a few that can, but most do not. Fermentation can work either in the presence of oxygen or in the absence of oxygen. It doesn't use oxygen. And then you don't need to know this one right there, generation of ATP, but you should know that aerobic respiration is the most efficient. You generate 36 to 38 molecules of ATP per molecule of glucose. Anaerobic respiration is the next most efficient, more than 2, so about 4, but fewer than 38. The highest is 36. Anaerobic respiration. How many ATP you make depend on the form of anaerobic respiration. And then fermentation only generates 2 molecules of ATP. Any question about any of that? Let me go back. All right, if there's no questions, I'm going to end it here, and I'll see you in the lab at 6:30. Any questions? All right.